Topic 12b, the Mendelian dihybrid cross. In the previous video, we considered a Mendelian cross with a single trait, which is also called a monohybrid cross. In it, we followed a single trait with two alleles across 3 generations and saw how the alleles will segregate from one another each generation. Next, let's consider a dihybrid cross, meaning 2 traits. In this case, flower color and height. Flower color will be determined by a single gene, denoted by the letter F, and represented as residing on a pair of chromosomes in each parental plant. The height trait will also be determined by a single gene, denoted by the letter T, and represented as residing on a different pair of chromosomes. If we cross-pollinate the parental true breeding plants, allow the seeds to develop, and then grow the plants, we would notice that all of the F1 generation resembles one of the parents. So the purple flower and tall traits are both dominant. To determine the genotype, let's consider the genotypes of the parental plants. Each parent has only one type of allele to give, so the F1 must be heterozygous for flower color and for height genes. All the F1 are dominant for both traits due to the presence of one dominant allele for each. The F1 cross will be a cross-pollination between the 2 F1 individuals who are heterozygous for both traits. Some of the progeny from this cross will resemble the original parental types. Some of the progeny will also resemble neither of the original parents and are called non-parental. When all the progeny are counted, we would notice that 9/16 would be parental dominant, 1/16 parental recessive, 3/16 non-parental that are recessive for the flower trait but dominant for the height trait, and the final 3/16 dominant for the flower trait but recessive for the height trait. Overall, this makes a 9:3:3:1 ratio. To determine the source of this ratio, we must first consider all the possible combinations of alleles that could be made from 2 heterozygous genes on different chromosomes. Both dominants could be paired, or one dominant and one recessive could be paired in more than one combination. Or both recessives could be paired. Once we know this, we can use a Punnett square to predict all the possible combinations of alleles in the next generation. Since there are 4 combinations of alleles possible from each parent, there are 16 possible combinations in the F2 generation. And we can see how 9/16 of the combinations are dominant for both traits. 3/16 dominant for flower and recessive for height, 3/16 dominant for height and recessive for flower color, and 1/16 recessive for both traits. To review, a Mendelian dihybrid cross follows 2 traits across at least 3 generations. If the genes responsible for the 2 traits are on different chromosomes, then they will independently assort, resulting in a 9:3:3:1 phenotypic ratio.